So DNA is the instruction manual of cells, and cell-free DNA, as the name implies, is DNA that's been released from the cell. And in terms of cancer diagnostics, we're typically very interested in cell-free DNA that's present in the blood. Cell-free DNA would be any DNA molecule that's been released from any cell. Circulating tumor DNA is a form of cell-free DNA where the DNA is derived from a tumor cell. So cancer cells in a body occasionally will die. They'll release their DNA, and that DNA can enter the circulatory system and be present and detected in blood. A blood sample could be used to identify cancer-associated mutations. The initial presence of those mutations would indicate that a cancer and cancer cells are present. Certainly, I think there's really tremendous promise that we'll be able to detect cancers more readily through blood tests. There's a lot of enthusiasm about an area of research and an approach called liquid biopsy. So this is rather than the traditional approach of taking a tissue sample from a tumor in a patient, one can monitor disease through the blood. And so it's safer and easier to collect, and it's possible to do repeated sampling before there's obvious disease, and then also during the course and after treatment. I think this is a really exciting time for cancer research. It's clear that these approaches are extremely promising, but there's still a lot of fine-tuning and a lot of specialization needed to make these effective for use in the clinics. And so supporting this research, it's really at a critical stage, and efforts now to push the science forward really I think are going to have long-term clinical impacts.